So again, mitosis is nuclear division. That is when the chromosomes are going to be separated. Those chromosomes that have now been duplicated, they need to get separated into the two new daughter cells. So these 5 phases are the ways that the cell does it. And again, I'm sorry I misspoke before. I said 18 hours for mitosis to occur. I knew that wasn't right. It's about an hour to 80 minutes. So it takes just a bit of time, but it's a pretty efficient process. Okay, the first part of mitosis is prophase. This is when that squiggly chromatin actually starts to condense into those chromosomes that you can see, those X-shaped chromosomes. And the centrioles, which are the mitotic spindle organizing centers, they're going to start to separate to either side of the cell and get ready to pull those chromosomes apart. We do have centrioles in animal cells, but there's a little different mechanism that you won't be responsible for knowing in plant cells, but just know that only animals typically have centrioles. All right, so the protein fibers that extend from the centrioles, we refer to them as mitotic spindles or spindle fibers. The spindle fibers that attach to the chromosomes are known as kinetochore microtubules, and I'll show you why in a second. Another thing that happens during prophase is that the nucleolus, which is inside the nucleus, that's the organelle that makes ribosomes, that's going to break down and disappear. Our prometaphase is the second phase of mitosis. This is when the spindle fibers actually attach to the centromeres. The kinetochore microtubules, as you can see, there is a arrow there in that picture that's pointing to the kinetochore of the chromosome. The kinetochore is actually the proteins that are on the outside of the centromere where the spindle fibers are actually going to attach. So the kinetochore microtubules are going to attach to those kinetochore proteins and make sure that the chromosomes are separated properly. The chromosomes are going to start moving during prometaphase. They're going to start moving towards the center of the cell. When they get to the center of the cell, it's actually metaphase, but they do begin moving here during prometaphase. Another feature of prometaphase is that the nuclear membrane breaks down. Okay, during metaphase, you can see now that the chromosomes are aligned along the middle of the cell. That's referred to as the metaphase plate. The kinetochore microtubules at this point are all attached to the kinetochores, which are those black dots on either side of the purple chromosome. And there's actually a checkpoint around this time, it actually happens in anaphase, to make sure that all of the kinetochore microtubules or the spindle fibers are attached to the kinetochores of the chromosomes. Otherwise, the chromosomes will not separate properly. All right, during anaphase, this is the fourth phase of mitosis. This is when the sister chromatids are actually going to separate and they're going to become their own new chromosomes. The way that the sister chromatids separate is actually those cohesin proteins that were holding the centromeres together. They break down due to an enzyme called separase. Separase enzymes degrade the cohesin proteins holding the centromere together and allow for those chromatids to be pulled apart. This mechanism is to ensure the same exact genetic material is getting to either side of the cell so that the new daughter cells will have the proper chromosomes that they need. The way that the chromosomes actually separate is using the 2 different microtubules that are in the cell. The kinetochore microtubules are going to shorten towards either pole of the cell, pulling those chromosomes apart, and the polar or non-kinetochore microtubules, so the spindle fibers that aren't actually attached to chromosomes, they're going to use proteins called kinesin and dynein motor molecules to move across each other, and they're going to actually lengthen and they're going to help push the cell and lengthen it so that the chromosomes can get to either side of the cell. Here's the separation of the chromatids in a little bit more detail. You can see the kinetochores are those kind of like the kinetochores, on either side of the chromosomes, and you can see that the microtubules are attached to those kinetochores. And like I said before, an enzyme called separase, how cute, it separates chromosomes, degrades the cohesin proteins and allows for chromatids to separate from one another. Oops, excuse me for that sound, that's annoying. Okay, so here's a chromosome. At this point, it's 1 chromosome, but 2 sister chromatids, and you can see the kinetochores right there are those blue ovals. And during anaphase, when they separate, excuse these sounds, I'm so sorry, I didn't realize they were still in here. They're going to separate during anaphase, and they're going to become 2 separate chromosomes. So we no longer refer to these guys as sister chromatids. They're actually 2 separate chromosomes now. and they're referred to as daughter chromosomes. Okay, the last step of mitosis is called telophase. Telophases are going to occur when the chromosomes have separated to opposite sides of the cell. The nucleolus you're going to start to see reform. The nucleus is going to start to reform. And because the process of cell division is almost over, the chromosomes are going to start to relax back to chromatin. During telophase, cytokinesis also begins. Cytokinesis is complete with the cytoplasm division is complete when there are two separate new daughter cells.